This protocol facilitates the preparation and administration of a virulent phage cocktail against Pseudomonas aeruginosa in zebrafish embryos.
The main advantage of this technique is that it allows an in vivo assessment of the phage efficacy in counteracting the bacterial infection.
The phage cocktail is efficient in both wild-type and cystic fibrosis zebrafish models, opening the possibility of applying phage therapy to bacterial infections in cystic fibrosis patients.
Phage therapy can also be applied to counteract specific bacterial infections in other model systems.
To prepare a phage stock for the experiment, add 1.25 times 10 to the 7th phages to an OD600 of 0.05 P aeruginosa culture to a multiplicity of infection of 1 times 10 to the negative 3, and incubate the culture for 3 to 4 hours with shaking until the OD600 drops to 0.1 to 0.3. At the end of the incubation, incubate the lysate with 1 microgram per milliliter of DNase and RNase for 30 minutes at 37 degrees Celsius, and pellet the cells by centrifugation. At the end of the centrifugation, filter the supernatant through a 0.8 micrometer pore diameter, and add 58 grams per liter of sodium chloride and 105 grams per liter of PEG 6000. Incubate the solution at 4 degrees Celsius overnight. The next morning, precipitate the phage by centrifugation. At the end of the centrifugation, remove the supernatant and carefully dissolve the phage.